Hello, my name is Mörth, and together with my co-authors Ingfrid Haldorsen, Stefan Bruckner and Noeska Smit, I would like to present you ParaGlyder. ParaGlyder is about probe-driven interactive visual analysis for multiparametric medical imaging data. So to the introduction. We are working here, for example, with endometrial cancer patients. Endometrial cancer is a type of gynecological cancer, and it's the most common cancer type in women in industrialized countries. So what you can see here is the Norwegian Cancer Report of 2018, and what we can see in this report is that the survival rate is going up, which is good, but also the incidence rate is going up. So we're dealing here with a really important problem. What are the radiologists facing now in terms of problems here? So to analyze the endometrial cancer, they're using multiple modalities or multiple sequences in the MRI. So at the top row, you can see the standard sequences that you use in MRI, for example, T1, T2, or diffusion-weighted imaging. And in the bottom row, you can see pharmacokinetical models. So these models are calculated out of a dynamic series within the MRI. So they're co-registered by nature, and they're showing different physical properties of, for example, the tumor. So you can see, for example, the blood flow, or you can see parameters showing how the contrast agent is going into the tumor, how good the uptake is, and how long it takes, for example. So what challenges and goals are now these radiologists facing, and why do they need our help? So one point is they would like to find visual tumor characteristics. So currently there is no clear, there's no clear property now how the tumor is looking in the different MRI characteristics. So there are notions, there are intentions, but they don't, but it's not like fixed yet. So they want to figure it out, and this can be done in an explorative way. So they also want to explore the inhomogeneity of the tumor. There's this notion that inhomogeneity of the tumor could be linked to aggressiveness, but this is still an active field of research. So they would like to explore if this is the case. They also would like to compare within patient and between patients. So this should also be possible. And the last thing, which is also challenging for the radiologist, is the number of modalities. So 5, 7 or whatever modalities is a quite high number, and it's not so easy to compare them anymore if you just have, for example, slice views. So to the medical background, this endometrial cancer is a cancer which is within the uterus, within the endometrium, and when we have a look here, for example, at the blood flow, it could one possible question could be, how is the value distribution within the circle that we drew here? But they are not only interested in this one circle in the blood flow, they're interested in many circles in all, for example, here 5 modalities that are present. And this circle is not fixed, they cannot tell now which area within the cancer is interesting. They want to move it around and explore the tumor interactively. So first we had a look at the related work. So there's, for example, one paper by Malik and others, and they provided us with a method to display up to 5 modalities within one single view. Then there's the work of Sergei Stopper, so he's a colleague of mine here in the university in Bergen, and they present us with a method how to display temporal data, temporal abstract data on top of spatial data. And we also looked at the visual analytics hypothesis formation application by Daniel Jönsson and others, and they also compare abstract with spatial data there. So why does our application differ now? So in our application we show spatial data in an abstract form on top of spatial data, for example. We also provide probing interaction in a 3-dimensional space, and we also visualize a similarity visualization. So in our requirement analysis, we did not only focus on gynecological cancer in imaging experts, we also worked with neurological imaging experts and with machine learning experts. So the machine learning experts, for example, would like to segment the tumor automatically. So after exploring with them what challenges they face, in the end we could identify 5 requirements. So there's requirement 1, comparing regions. The radiologist told us it would be interesting for them to compare regions, for example, within the tumor and outside of the tumor, but also within one patient and then compare it to another patient. Then we have requirement 2. multiple modalities in a single view. So this is really challenging. As we've seen before in the related work, we had up to 5, but in our case we can have up to 7 or even more modalities. And the radiologist told us they would like to have it in a single view so that they could have a nice overview. Then we have requirement 3, the similarity analysis. So there's this idea that metastases which are in the same volume as the tumor maybe have similar properties, and these metastases could show up when we analyze similarity. Similarity could also be used to visualize the tumor or to segment the tumor. Requirement 4, parameter selection for multiparametric segmentation. So this is primarily a requirement of the machine learning experts because they would like to use multiparametric segmentation, and therefore they would like to know which parameters are most important to differentiate the tumor from the region outside of the tumor. And in the end we have requirement 5, an inhomogeneity analysis. So they would like to see how inhomogeneous the tumor is because there's this notion that it could be linked to aggressiveness. So now to our method, the ParaGlyder. Hey, I said ParaGlyder, not drone. Could you please move? ParaGlyder. So first out we start with spatial data. In the spatial data that you can see here, we place probing spheres. With this multiparametric probing spheres, we can then visualize the data in a non-spatial way, for example in our case in a radar chart. So we chose a radar chart because this shape can be recognized again in our glyph design. So what you can see here is the comparison between one patient and another region within this patient, but we can also have multiple patients being compared in this view. So this information can be then used for a similarity calculation, and we end up with the similarity volume. And this similarity volume here nicely shows the tumor and the metastases segmented. And in the middle we have our approach to visualize multiple parameters on top of a single view, which is called the Rixels, which stands for radar chart pixels. So now to a probing. Now we really have this ParaGlyder, and this ParaGlyder now goes into the volume, and when it glides around in the volume, you can see the radar chart updating automatically. So what are we showing here in this radar chart? The radar chart consists of 5 or even more modalities, and the axes are lined up in a star pattern. And what we're doing here is we take every voxel within this sphere, we're calculating the mean and the quantiles, and the mean is a dot on this axis and the quantiles are the band that you can see, for example, in K12. And what you end up is something like a radial box plot showing us the data within the sphere. And this is how it looks like in the application. So we are probing the volume, and we can also adapt the size of this radar chart, and as we're moving around the probing sphere, the radar chart is updating automatically. With the band we can also see if the data is inhomogeneous within the sphere. Then we can adapt the size. As we add a second sphere, we can see now these two spheres in the radar chart, and we can compare their results there. To give the user the full control over this probing, we have several interaction methods. So one is you can resize the sphere. Another one is to move the sphere freely around in the volume. But to really precisely locate the sphere at a specific point in the volume, you can, for example, fix the XY plane or fix the Z plane to really precisely put it where you want to have it. And for the neurological imaging experts, we provide the mirroring function. So when they place the sphere in one hemisphere within the brain, the second sphere is mirrored to the exact same position at the other hemisphere of the brain. So comparing regions, check. Now on to the Rixels. So the Rixels is our approach to visualize multiple modalities within one view. So we're laying a grid on top of our modalities, and the modalities are nicely aligned. And within each of these grid cells, we're probing our modalities, all of them. And then we do a similar approach to the sphere that we have seen before. So we're taking all the values within this one grid cell, we're calculating the mean and the quantiles again, and then we produce the radial box plot. But instead of showing this box plot, we're taking now only the geometric object that is prescribed with the mean values connected. And this is our glyph that we present within this grid cell. And this is how it looks like in the application. So this is our Rixel view. You can see the grid, and in every grid in the middle we are presenting our Rixel. When I hover over the Rixel, you can see a tooltip showing the radial box plot. We can adapt the grid size so that the grids are getting smaller and smaller and we're getting a more detailed view. We can also adapt the Rixel size. We can then change the slice that we want to look at. And we can also crop the volume so that the Rixel view is only showing us the tumor, for example. So we got the more detailed information on this particular region. And we can also select which modality we would like to see in the background. So it could be the blood flow, or it could also be other parameters that the radiologists are interested in. So multiple modalities in a single view, check. So now on to the similarity view. In the similarity view, we're using the probed information of the sphere, and we calculate for each and every voxel within the volume how similar it is to this multiparametric characterization. And then we end up with a value per voxel which we can display using a color map, for example the Viridis color map. And as we do so, you can see here that the tumor and metastases are highlighted. So for the metastases, we have to say it can be that they are in the same volume as the tumor, but this does not have to be the case. But if they are there, we have seen in this one example that the metastases are highlighted. And what is the benefit now? What hasn't been possible before? The metastases are highlighted. So when you look at the normal modality as seen before, they were not so clearly visible. So this is a real benefit for the radiologist here. So similarity analysis, check. What else can we do with the similarity? So for the similarity, you can also select which parameters should influence the calculation of similarity. So with this, you can then decide which modalities are the most important. So for example, which combinations do still give a clear segmentation of the tumor and the metastases, and which combinations, for example, are not showing anything at all. And this is very important for the machine learning experts, because they gave us the feedback that they, for example, would like to decide prior to the machine learning which modalities are interesting for the segmentation. So parameter selection, check. The similarity can also be used to color our Rixels. So by coloring them, we can see then how homogeneous the tumor is. So this is an example for a quite inhomogeneous tumor, and this is an example for a quite homogeneous tumor. So you can see the Rixels are getting more yellow, more similar they are to our probed region. So inhomogeneity analysis, check. Now to the implementation. So we used VTK-GS for all the medical images, and we used D3 for all the non-spatial information visualization. Beside that, we also used JS3 for the patient and data visualization and interaction, and we used jQuery layout for the window layout. Now to our results. So for the probing, we can see a result for one patient where we had one sphere in the tumor and one sphere in the normal endometrium as a compare region. And what we can see here is that some modalities have an impact. So for example, the K12, but the blood flow, for example, there both spheres have quite similar values. So this gives us a nice information about which modalities are interesting for segmentation tasks. Beside that, we can also compare different regions within one patient and another patient and see if these characteristics are the same for multiple patients. This could then be used, for example, for a cohort analysis. For the Rixels, we can see here examples for homogeneous tumors and inhomogeneous tumors, and we can also in the tooltip then nicely see what values are used for these Rixels and what are they presenting. To the similarity, we see here different data sets. So we have one data set for endometrial cancer and we have one data set for neurological imaging. And we place the sphere in endometrial cancer within the tumor and in the brain within an inflammatory region. And for the tumor, we can see that the tumor and metastases are highlighted. And for the brain, I would like you to focus on the skull and the inflammatory region where the blue sphere is located. And if you would like to use only this one modality to segment, it would be difficult because there are multiple regions which are looking similar in intensity. But with our multiparametric segmentation, we can see that only the inflammatory region is highlighted. During our project, we were working closely together with 3 experts, one of each field, but in the end we were really lucky that we had 11 experts evaluating our tool together with us. So the evaluation started by presenting them the application and giving them a short introduction to it, and then afterwards they were able to explore the capabilities of our application, and afterwards they could give us feedback in general and more specific statements, how useful they find the application and what was difficult for them. So to our evaluation result, you can see here that there are general statements to the left and more specific statements to the right. And green means it was good and red means it was not so good. So we have a lot of green in there. The experts really like the application and they find it particularly useful. They find the probing really useful and also the Rixel view. Then the similarity, there was almost no negative feedback about that. And in the more specific statements, we also got the response that they find it really useful and they would like to work with us together more in the future and to use it also. So what are these red spots? Now here I would like to give you a short notice on them. So we developed this application for research purposes, but we wanted to know from the experts also how useful they would find it in a clinical routine. And there we could see that some say, yeah, that could be useful, and some said, I'm not so sure. So to give you more input on that, in a clinical routine, radiologists have to read images quite fast. It could be that they only have 15 minutes to read one data set of a patient, which is quite short. So for the application to be useful in the clinical routine, it either has to make their work quicker or it really has to have a benefit for the patient. And our experts don't know that yet. So we have to look into more detail if this is the case, and it could be that maybe some parts of our application go really to clinical routine. Yeah, and we also conducted the system usability scale to make our results comparable to those of others, and we got the result between good and excellent. So now to our lessons learned and some limitations of our applications. So we have seen, for example, that there are strong opinions on interaction methods. So we had a look at radiology setups and how they interact with the setup, and we tried to give the radiologists the interactions that they're used to, but during the evaluation we have seen that this is not standardized. So some radiology experts use different tools than others, and for example the slicing could be by scrolling but also with a slider, so this is not standardized. So here we have got the feedback, okay, we should for example make it customizable, that we say, okay, this is for example for neurological imaging and they're used to that. And for endometrial cancer radiology, for example, they're used to something different. So this should be customizable. The path to clinical routine implementation is tough, so there are lots of steps, and it could be that only some parts of your application are usable in the end. The evaluation demonstrates benefits and feedback, and this gave us a strong feedback where we can adapt future applications more specifically to the needs of our experts. When looking at the comparison between multiple patients, we see a possible limitation. So it's usable maybe for 5 or 6 patients, but if you go with a higher number, you have to think about how to visualize that. Also, one problem with this comparison is you cannot place the sphere exactly in the same region in every patient because every tumor is different. Also, we can only work with co-registered multimodal data. For the endometrial cancer, we had these pharmacokinetical models. They are co-registered by nature. We also had neurological imaging methods, which also were presented in a co-registered way. But it could be that you work with not co-registered data, so you would have to do this pre-processing step. Now to our conclusion. So we present the probe-driven interactive visualization, where we display spatial data as non-spatial data visualization. We present the Rixels, which displays multiple modalities in one single view. We calculate similarity by Euclidean distance and have seen that it highlights the tumor and the metastasis, which has not been possible before. And we conducted an evaluation with 11 clinical experts. And for the future, we would like to take the characteristics that the radiologists find using our system to a cohort level and see if this characteristics holds up for a higher number of patients. And this could also be used then, for example, for segmentation purposes. Thank you very much for your attention. I'm ready to take off. But first, I'm looking forward to your questions.